So this is chapter 13 in unit 7 on meiosis and genetics. So we're going to start by taking an in-depth look at meiosis and comparing it to mitosis. So the two main types of reproduction are asexual and sexual. And with asexual reproduction, you have a single individual or a single parent who is the only parent and therefore will pass all of its genes on to its identical offspring. We have no fusion of gametes, but we do have mitosis and meiosis involved. And then with sexual reproduction, we have two parents passing on each of their sets of genes to their offspring. And then the offspring, therefore, will be varied genetically from their parents. They will not be identical. And this occurs through meiosis and fertilization. So, two definitions before we go any further is one, heredity is the transmission of traits from one generation to the next, and variation is demonstrated by the differences in appearance or the phenotype that offspring show that is different from their parents and different from their siblings. So in asexual reproduction, we have mitosis occurring, and the way that asexual reproduction is going to work is through mitosis. You're going to end up with a daughter cell that is identical to the parent. When we say we have an exact copy, we say we have a clone. So in asexual reproduction, we are producing clones. These clones, these daughter cells or these offspring, have the same amount of DNA as the parent, the same number of chromosomes, and the same genetic information. And the only way that you end up with any variation with asexual reproduction is through random mutations that occur during the mitotic process. So some examples of asexual reproduction, we have single-celled eukaryotes or simple multicellular eukaryotes. So with a single-celled, or an example of a single-cell eukaryote would be like a yeast or a fungi in the fungi family, and also protists like parameciums or amoebas. So each of those reproduce through asexual reproduction. And then some examples of multicellular eukaryotes are things like hydras, which we did back in our species interaction lab. We use little hydras, which reproduce asexually. So whenever we're looking at life cycle or how reproduction occurs within a species, we want to ask questions of such as, what are the disadvantages or advantages of this type of reproduction? So when we were studying plants, we looked at some of the advantages and disadvantages of both the sexual and asexual reproduction options that plants have. And it's similar with animals as well. So some of the disadvantages, or probably the biggest disadvantage, is that we lose any variation in the genome that's being passed down from parent to offspring. And therefore, there's more susceptibility to disease or mutations that are actually harmful. So we like to get genetic variation for the health of a species. But the advantage, of course, is not needing to have close proximity for reproduction to occur with another parent. And also the advantage that you can have more frequent reproduction and more efficient reproduction. And also you have the advantage of not having competition for mates, so we don't have resources being wasted with some sort of intraspecific competition. So this was a video showing the budding of hydra, but we're actually not going to look at that. But you can find YouTube videos that will show you this, but it's really just what you would expect. Through mitosis, you have a hydra growing off the side of the parent and then just budding off. So let's turn to sexual reproduction and talk a little bit about that. So if we have a complex multicellular organism wanting to reproduce, in order to do so, we have to have the joining of egg and sperm. And egg and sperm are what we call gametes. And these cannot be made by mitosis, because if they were, we would end up with a problem that looks something like this. So in a human egg, our human cells have 46 chromosomes. So if a human egg had 46 chromosomes, and a human sperm also had 46 chromosomes, just like all the rest of the cells in the body, when they fused to make a zygote, we'd end up with 92 chromosomes. So of course that doesn't work. So when we're looking at these different options of either sexual or asexual reproduction, what we want to keep in mind is that a life cycle is the generation to generation sequence of stages in the reproductive history of an organism. Some important facts that you want to also keep in mind is that children do not inherit physical traits from their parents. And we're going to look at this even more in depth when we're looking at evolution and adaptations in fitness. But it is the genes that are actually being inherited. So when we're talking about heredity and looking at genetics, you're going to need to keep that in mind, that children are not inheriting traits from their parents. They're inheriting genes. So genes are the units of heredity, and they're made up of segments of DNA. And genes are passed on to the next generation through the reproductive cells, the sperm and the eggs that we called gametes. Most DNA is packaged into chromosomes, and the reason that we say most DNA is because you need to remember that mitochondria and chloroplasts also contain little bits of DNA. But most, as you know, is located within the nucleus in the tight little packages of chromosomes. And humans have 46 chromosomes in each of their somatic cells. Somatic cells are another way to define those are just the body cells. So every cell in the human body except the eggs or sperm, which are the gametes, have 46 chromosomes. And a gene is actually a portion of a chromosome, and its location on that chromosome is called its locus. So looking here at this diagram, you can see that this is the chromosome, and it consists of a single DNA molecule that's a series of nucleotides joined together. And this single DNA molecule also has various proteins attached to it, which we call histones. And each chromosome, such as this, contains hundreds or even thousands of little portions of the DNA that are defined as genes. And where these genes are located, again, are called the locus for that gene. This is what is called a karyotype, and it's a picture of a set of genes, and it's an ordered display of the pairs of chromosomes from a cell. So how this is obtained is a picture is taken of chromosomes during metaphase. So they apply dyes, and the dyes actually will show the unique banding of similar chromosomes. So if we look at this here again, try to advance this, you can see from that picture, they'll match up the chromosomes into pairs. And each pair of chromosomes are called homologous chromosomes. And so in, again, every somatic cell in the human cells, we have 22 sets of homologous chromosomes, which add up to only 44 chromosomes. So the extra 2 that make up the 46 are what we call the sex chromosomes. So we can tell that this is a female because there are 2 homologous sex chromosomes which make this a female. They're both an X chromosome. Now we'll compare that next to a male set of chromosomes. So in this case, oops, where'd it go? Well, I need to go back and find out what happened to the male set of chromosomes. So hold on just a second. Okay, so this is a karyotype of a male set of human chromosomes. So you can see here we have again our pairs of homologous chromosomes, and again homologous chromosomes are about the same size, and so you can see how they match up, but more importantly they have the same banding patterns, and those bands show that they're actually carrying the same genes on each chromosome. So that's how we can tell, for example, these are fairly close in size, but the banding patterns here are similar, whereas here they're quite different from these. And we can tell that this is a male set of genes because we can see our 22 pairs of autosomes, which are our homologous chromosomes that are not sex chromosomes. So we stop here at 22, and when we look at the set of 23, we can see that they are not homologous. They are not the same size. They are very different. And that tells us that here we have a male with one X chromosome and one Y. And these are not homologous. Again, they're not the same. And you can see that the X chromosome carries a lot of genes, and the Y chromosome is only this size. It's much smaller and does not carry nearly anywhere near the same number of genes as the X chromosome. Okay, let's see. There we go. So what we were looking at before is one type of karyotype, and that's actually the original type of karyotyping that was done. And nowadays, fluorescent dyes are used to actually map out sets of chromosomes. So you can see here, they're still dyed during metaphase, but they're dyed in a way that we have much different colors going to each of the different, and it makes it much easier actually to pair up, because the different chromosomes will actually pick up different colors of dye, a fluorescent dye. And again, you can see that this is a female because we have 2 X's that are about the same size, and we still see the pattern with 22 homologous sets or pairs of chromosomes. These 22 again are called autosomes, and then these are called the sex chromosomes. This is another picture showing a close-up of how the fluorescent dyes pick up a lot different views of the different chromosomes, but you can see how the shapes are very similar with the homologous pairs. And this is a good time to actually point out what are we looking at when we look at a homologous pair, because it's important to remember, what makes up a person's set of chromosomes. And the reason that we end up with two that are carrying the same